Hello, my name is Cheng. I am a PhD student of Institute of Biomedical Engineering, University of Oxford. My project aims to neuromodulate the hippocampus non-invasively through the use of transcranial ultrasound stimulation. Here's a quick update showing the works of simulations that have been done. Here is an overview of the project. The bilateral hippocampus structure are going to be targeted using two ultrasound transducers simultaneously. The project consists of 3 parts: simulations, experiments, and the analysis of the human trial results. The simulations are done in K-wave 2 box using human participants' MRI scans. Desired source conditions, resulted pressure distributions, and thermal distributions can be estimated inside the simulations. The experiments are going to validate the findings of the simulations, and then the neuromodulations on human participants will be conducted. By analyzing the data from the human trial, stimulation effects will be revealed. Here's an example of the MRI scans. The red crosses shows the marked targets by Dr. Chris Butler, Imperial College London. Target 1 and 2 on the left are the target sites of perennial cortex. Target 3 and 4 are sites of anterior hippocampus. Here, the workflow of the simulation procedure is shown. The MRI is segmented into air, skin, skull, CSF, gray and white matters. The segmented data is updated from the resolution of 1 mm of the MRI to 0.5 mm grid size of the simulation. This grid size used in simulation is determined through an analysis balancing between the computational cost and the accuracy of the acoustic simulation result. The time reversal simulations is performed by putting a virtual point source at the target and the ultrasound radiates outwards. A plane sensor is placed outside the head, covering the potential positions for placing the transducer for stimulating this target. This sensor plane records the phase and magnitude from the time reversal simulation. The recorded phase is used to focus ultrasound at the target for stimulation effects propagating through the inhomogeneous medium. The recorded magnitude is used to determine the optimal placement of the transducer inside the sensor plane for maximum power transmission through the skull. Finally, forward simulation is performed, revealing the resulted pressure distribution inside the head. Using this pressure distribution, thermal simulations can also be performed. Therefore, safety criteria can be ensured before stimulating on human participants using the simulation results. The simulations are performed, and as an example, in one volunteer, the left perennial cortex and anterior hippocampus are stimulated in 2 separate simulations. The resulted pressure distributions are overlaid in this figure. This figure shows that each target can be stimulated with high pressure separately at a time without introducing high pressure at the other target on the same side. To implement the phases for creating the focusing at target site in reality, acoustic lenses are needed. The lenses are decided to be made of PDMS material. Simulation with PDMS lens was performed. The resulted pressure distribution are shown in A, B, and C. These 3 orthogonal views passing through the target site shows a tight focal spot with high pressure gain exactly at the target site. The -6 dB focal volume is around 30 mm3. Figure D shows the surface outline of the PDMS lens structure used in this simulation. Further analysis also determined the desired diameter of the transducer to be 65 mm and the frequency of the ultrasound to be 500 kHz. Future works will be conducted to validate these simulations through experiments on an ex vivo skull sample. Thank you very much.